All right, hello everyone. I'm Josh Ives, and as Ms. Holberg just said, I did computational biology at CMU. And so computational biology is a pretty wide-ranging topic, and it covers a whole bunch of stuff that's basically all biology. So I'm just going to kind of highlight 3 different things that I did while I was there. And if I start just talking at you really quickly, feel free to ignore me. It'll maybe happen. So the first thing that I learned about relatively early on in the internship was genome assembly. And this is kind of the process that we use to actually figure out what the genetic code of anything is. You can't just exactly run the entire DNA strand through a reader. You have to break it up and then try and reassemble it. And my mentor had a really good, one second, sorry. Oh, and there it goes. Okay, my mentor had a really good analogy that was you take a stack of a newspaper and throw a stick of dynamite under it and then just pick up all the pieces once it's done and try and reassemble it. This is really difficult to do. They tried to do it for the Human Genome Project for something like 10 years, and it turns out if you use computers to do it, it's a lot faster. So that was a large part of what I spent my time learning how to do. And then second project I was really involved with, probably took up the most of my time, was something involving cell imaging, where I was looking at cells under a really fancy microscope and tagging them with different dyes so that I could actually see what was going on. In this case, if you want to read that, that's green for lysosomes, red for mitochondria, and blue for DNA. And we wanted to get a whole bunch of images. I apologize for my face there. And so to do this in the volume that we wanted to do, we used a liquid handler, which is the machine behind my face there, and it just picks up liquids and moves them around and does a whole bunch of stuff so that you don't have to. So I learned how to use that, and I was able to get a grid that looks sort of like this, ignoring those back bits, where we put each of these wells had a bunch of different cells in them, and we put a whole bunch of different drugs at a whole bunch of different doses. Sorry, it doesn't actually matter too much what these are. Basically, we had 3 different drugs that should hopefully do something to something. I don't know. They'll do something in the cells, and we had them in different doses. And then we wanted to get a machine learning program to actually be able to distinguish between all of these different things. And so we set a microscope to work, and it did work that would have taken us weeks in about half an hour, and we ended up with more than 1,000 images. And we had a bunch of data to work with, and that was awesome. And actually using these images was really long, complicated process that I won't bore you with too much, but we had to basically convert the files from a useless format that no one cares about into something actually usable. We had to contrast search the images, which means you take them from being basically black to actually being visible so you can see what's going on in them. And then we applied a machine learning campaign to them to try and see if we can get a machine to actually understand what was going on with these cells. Because looking at them, and I looked at many of them, you cannot tell what is going on at all, but a machine can, we hope, tell the difference between the different types of drugs based only on the image it's seeing. And the ultimate goal was to get this kind of graph, which we did, and it shows, if we go here, that basically as time goes on, the machine learning program is able to determine more and more about what it's looking at. And so only having looked at about 25% of the actual data, it was able to classify with 50% accuracy. So it knew what it was seeing more of the time than just if it was guessing pretty early on. And then over here, we wanted it to identify, if you remember those 4 different drugs or 3 different drugs in a control and different doses, we wanted it to identify, distinguish between those, and it was able to distinguish between only 2 of the drugs, which is kind of unfortunate, but it was able to distinguish between all 3 of the different doses, which is really cool. We got a machine to understand what we wanted it to understand. And then the third thing that I'll talk about very briefly is Lactobacillus roseus. This is a bacteria that we sort of discovered in the time we were there. It's related to a bunch of these other bacteria that you've never heard of and that no one else has ever heard of. But it was a very cool discovery for me just because it's a basically new bacteria. They've only found it once somewhere else, and it's never been sequenced, that thing I told you about in the beginning. So that was what I got to do. My role in determining this bacteria was basically I'm going to get a bunch of that bacteria by growing it in various conditions. I'm going to extract that DNA for sequencing using a complicated, weird chemical process where I just follow instructions. And then I tested for antibiotic resistance to tell us a little more about that bacteria and where it likes to live. And this is actually going to be a research paper, which is insane. I'm a high school student, and like, I don't get a chance to say this at the end, but I just really want to thank Joshua Kangas, who was my mentor. He let a random high school student come into his lab and help do a research paper, and that's amazing. So in my free time, I did a bunch of stuff around Pittsburgh, where CMU is located. I went to Phipps Conservatory, which has got all these cool glass sculptures in the midst of a huge botanical garden. That's also where you see this cool little tree. Went and explored the campus at CMU. They have this cool Rainbow Bridge and a bunch of other excellent buildings. Went to amusement park, which I didn't have time to put in here. And I just got to explore the city of Pittsburgh, which was really, really cool. And I'm super glad I got the chance to check it out. My advice for future Pinterns would be really proactive in searching for something. Like, Sarah will find you a thing. She knows everybody, and she can find something. But if you actively look for and really try to find something that you will enjoy, it's there somewhere, and there will be people willing to take you and do something really awesome with it. Thank you so much.